So, right, good evening, students.
Good evening, sir.
Good evening. So, how did you guys write the test? Did the test go well? I see that 4 of you have submitted them. Vishal is not here today.
I felt that I lacked confidence. I don't know.
Okay, it's all right. So, I think the test is not to prove how much you learned. I think this test is about how different is a ball game of writing the exam, especially in the physical anthropology part of it. Because here we are trying to figure out what are we missing, what components are needed, is your preparation, is writing style, timing. So we are trying to figure it out. So don't worry, don't try to judge yourself now. Okay, just do it and see how it went. Okay, right. So anything specifically that you want me to tell? Are the questions doable for you? The question should be straightforward. Am I right? So this is most of the questions that we picked up from the past exams. Some of them, just to review, they might have not appeared in the exams before because we have to cover different aspects of the unit. Otherwise, questions are as asked in the exams before and covering different dimensions. So you felt them, they are okay? Questions are okay? Timing?
One question:
Sorry.
the synthetic, how it integrated the Mendelian and genetics, and so I wrote something, but that is what I only could get. I was not at all confident in that.
That's all right. That's okay. Because we will, I'll see. So first I will give you the feedback on it. And then we will have a quick discussion, maybe during a session like this. We'll quickly go through the structure of the questions just to highlight the common mistakes you have done. Okay, I'll give you individual comments and then I will discuss together covering different mistakes that you have done and what is the best way to handle those answers. Okay, then you'll get an idea on this. We'll do that sometime this week. Uh, anything else that I need to know related to exam? Okay, if not, so can we plan another test on coming Sunday? Do you have any test planned for Sunday? Can we plan for a test?
And we have physical, so
Social.
cultural also. Yeah.
So do you want to write a test before or after? When can we plan the next one? Because we
Definitely after.
Yes, sir.
Okay, right. So next units must be 9.2, right? 9.2, 9.2, 9.4. You want to write a separate test for each unit? 9.2 is small, so we definitely have to combine with something else. So instead of 1.7, I think, are you comfortable with 1.7 or do you want to write answers? Because questions are not coming from the 1.7 part, like cell division, DNA replication, just conceptual understanding enough. For the exam point of view, we are not getting questions on that. So it's up to you. If you want to write a test, I can give you a test, no problem at all. Or we can actually skip that and focus on the other units. Because we finished 1.4, we finished 9.3. So 9.2, 9.4 must be our next choice. Okay. 9.2 for sure completely. And if you want A, B, C, but yeah, 5 questions will be gone. Yeah, 9.2, 9.4 will do next. Okay, so we'll do after the Sunday. So can we have it in the mid of the week so that we can finish most of the tests by the time we're covering up the syllabus. So can we say something like a Tuesday or Wednesday next week after Sunday? Is that good enough for you?
Thursday maybe, sir.
Thursday. Yeah, sure. So 9.2 and 9.4. Okay, 9.2 and 9.4. Anyone who have not submitted the exam, still writing?
I have not submitted, sir. I am still writing. I will submit.
Okay, sure, sure.
On the portal or Telegram, sir?
Students have submitted on the portal. Let's do that. How are you doing it in the social part?
Both.
Yeah, it's all right with me. Any of them is all right with me. Okay. Only thing is, if you submit on the portal, I think I can get the idea about the trend of how you are getting scores. So preferably on the portal, unless you have any issues on that. So let's do on the portal. Next text will be on the Thursday, 9.2 and 9.4. Okay, start preparing, spend little time every day. Look at the past questions. Look at the past questions. So questions are known in a way to you. What we discussed in the class, what are the past questions? Focus on them. We are going to do those questions. Okay, right. So good. So in the last class, we finished talking about the twin studies part, twin studies, co-twin method, faster child method. Correct. Those topics we have finished in the last class. Okay, so today we are going to first begin with the biochemical genetics part or biochemical methods we have in the syllabus. So today we'll focus on biochemical methods and immunological methods. Okay, biochemical and immunological methods we're going to focus on today. Okay. So what is biochemical methods or biochemical genetics? Because usually if you see in the exam, once a question had come on the biochemical genetics, what do you understand by biochemical genetics? So given that in unit 9.1, we are focusing on methods to study humans. So far we focused on cytogenetic methods and twin study methods and the pedigrees. So they're working on different problems related to humans, correct? So here we are going a little bit on the molecular side, meaning at the level of molecules. Okay, so this is called biochemical methods. What are biochemical methods? What is biochemical genetics? First, we'll understand the scope of biochemical genetics, and then I'll pick up a couple of areas, couple of diseases in the traditionally that we study under biochemical genetics. And I'll give you two examples of methods and with a case study of how biochemical methods are used in dealing with diseases. Okay. Okay, I'm sharing the PowerPoint slides for that.
Sir, in twins, there are question and diagnosis of twins.
Come again.
In twins, there is question on how to diagnose the twins.
There's a question on how to diagnose the twins. Okay, you mean in the, like you're asking about in the questions that came in the mains before? Are you talking about one of the past questions? Can you read the question for me, please?
Define twins, describe the methods of diagnosis of twins. In what way are twins useful in the study of human genetics? 1998.
1998 question. Okay. Yeah, I don't remember seeing that question. That's what I was looking at. I don't think, because in these days, since even after that, we are definitely seeing the role and importance of twin studies, but how diagnosis of twins has happened has not been seen. Even if I have to think, I think usually ultrasound method is what is used for diagnosis of twins. Because I don't think people would suspect that. So one thing they would see is as soon as the diagnosis happens in the 5th month, that is about 20th week of the pregnancy, when they get to know about if there are two babies, and then there's a more greater deal of classification, whether babies share the same placenta, same amniotic fluid. We have something called chorionic layers. So more analysis can be done on that, but usually ultrasound, what is used in the 20th week of pregnancy to identify whether they're twins and normal children, because few complications related to that. Normally what happens if there are two twins, the amniotic fluid tends to decrease, and the deliveries, some complications are there with twins compared to normal deliveries, and the delivery may happen a bit earlier than usual. So some extra care has to be given or has to be taken when the twins are there. So, but otherwise ultrasound is normally the routine method for that. Okay. So I don't think I've seen that question even later in last several years, but twin studies related component can be asked. How is twin studies, how is it designed? Okay, and examples of how do we calculate concordance rate and the discordance rate. So if you focus on those topics, I think that should be much more than sufficient for the exam point of view. Okay, point one, genetic study, DNA technology, molecular anthropology, immunogenetics, twin method in human genetics. Short note that came in 2013, twin method in human genetics part. But these angles I just mentioned to you will be sufficient. So question may not come directly, but they may not ask you, because these days we are seeing that question may not be simply describe twin method in human genetics. They may ask you like this: nature, and discuss about nature and nurture debate. And with respect to nature and nurture debate, discuss the method that is routinely used in addressing this issue. Okay, question may come like that. They may not directly specify.
In this question,
Yeah.
what will be the answer structure in this question?
What should be the answer structure for
This question, the role of twins in nurture and nature debate.
So yes, I think exactly what we discussed in the last class. You first discuss what is nature nurture debate. So what is the role of nature? That is no role of genes, and what is the role of environment? Okay, you describe it briefly and say that genes or DNA forms a basic component for any characteristics for that matter, but together with environment, the outcome is defined. You discuss like that in the open discussion. Then twin studies have been used as a great tool in understanding about the role of nature or role of genes and environment. Then you say, in a following example, you can talk about one, you can give the features why twins are actually suitable for this. So you talk about monozygotic twins, dizygotic twins, and you say, given that monozygotic twins actually share the same genetic material, genetic components, so we actually assume that they have same genes. So any effects could be because of the environmental changes. So you write about the assumptions that we normally make with twins, monozygotic twins, and then see why we actually have to compare with dizygotic twins. You say how exactly we come across, or we come out with the idea about the role of nature or nurture by comparing with dizygotic twins. There, what do you say? You would explain that when we compare when we calculate the concordance value for monozygotic twins versus dizygotic twins, you will know if the concordance rate is so and so. If it is actually 100, close to 100% for MZ twins, and close to 50% for dizygotic twins, then it should be the genetics that playing the most important role here. But usually what happens, you will see the combinations of both. So like that you explain, give an example for that. Either you choose to write the case study that I have shown you, I shared you with the chapter, textbook chapter I've given you. Talk about that, the recruitments of military and their weight and height that changed over a period of time, and 77% of value for heritability value, or 0.7 value for the this indicating it is mostly nature. Like that you describe it. Okay, in the end, if you want, you can mention about the many things like schizophrenia I mentioned to you, or a couple of things that we talked about. So those type of characteristics and role of genes versus nature environment can be found. That's how it should be the answer. So basically what you do every time you're trying to answer a question, divide that question into sub questions and try to answer those sub questions in a logical manner and write it. Okay, this is a structure I think best suitable for such a question. We'll do that because I think that is something that we're going to try in the coming tests. So not next test, but test after, we're going to work on that. Is that clear? Yes.
Yes, sir.
Right. So you guys probably heard about biochemistry, biochemicals, right? people with no bio background, what comes to your mind when we think about biochemistry or biochemical methods? So any in day-to-day life, have you seen anything connected to biochemistry? Do you read it anywhere? Have you seen anywhere that you places that you visit? So what comes to your mind? Because this is one area where students, I feel they tend like they get lost. So just wanted to start with what you know.
Sir, tablets.
Tablets. Okay, right. So tablets have something to do with the biochemistry or chemistry? Which chemistry?
Biochemistry.
Okay, right. Where else? That's the only place that where you have seen biochemistry is done here.
Curd.
Curd. Okay, right. So Vishal, your background is
So I have some background in biology,
biology, right?
biotech.
Biotechnology, right. Shweta, have you seen biochemistry anywhere? So definitely something to do with the drugs, as we hear. Few more places that actually I'm sure you've seen it, but you're not able to recollect. Anything you would like to add, Shweta? No?
No,
Have you
sir.
gone to laboratories
Blood
for
tests, blood culture.
diagnostics done? Biochemistry is what you see. What is done in the laboratories, they mention that, right? Pathology, biochemistry, they would normally write the list of those areas, isn't it? So biochemistry is basically dealing with the chemicals in our body or chemicals in biological systems. Biochemicals. So these are also chemicals, but naturally found in our bodies or in bodies of living systems. So biochemicals. So as soon as you think about biochemicals, remember that. Normally, drugs don't come under biochemicals. Drug metabolism may come under biochemicals because drugs are purely chemicals. Drugs are purely chemicals. Okay. So organic synthesis or organic chemistry basically is the part of chemistry that we use for synthesizing drugs or analytical chemistry to analyze them. So drugs are chemicals. Okay. But these drugs actually, when they are participating or causing something or controlling something, when you take the medicine, like say you've taken antibiotics, when the antibiotics get into your system, what happens to that antibiotic? What part of our body does it deal with? That comes under biochemistry part. But synthesis and the creating production, that comes under chemistry part. Okay, right. So do we have chemicals inside our body? Are there any chemicals? Can you name a chemical in our body? Are chemicals in our body? Vishal, you will be the last one to answer this.
So dopamine, oxytocin.
Oh, oxytocin. Very good. I didn't expect that. Great. Dopamine and oxytocin. Excellent. Any other chemicals?
The bile juice.
Wow, very good. Bile juices. Okay. Right. Yash, it's your turn. Find something. Do you read something on the wall? No, what chemistry? What gives us energy, Yash? What gives us energy? What chemical gives us energy? Let me be more specific. There is a miracle chemical in our body which gives us energy. In the form that we can use, our cells can use them. Powerhouses, what do they produce? Powerhouses in our cells, what do they produce?
ATP.
ATP. Have you heard about ATP? Adenosine triphosphate. Okay, so it is a chemical, right? It's a biochemical. It's basically several adenines that we have in the DNA, like that with 3 phosphates, adenosine triphosphate we call. So it's packed with energy. When we break it, it's like eating munch or whatever chocolate you like. It's like that. So you break it, you get the energy from it. So like that, there are many biochemicals in our body. So in biochemical,
Nucleotides there.
sorry, yes.
Nucleotides there are. Carbon cycles.
Carbon cycle,
Nucleotide sequence.
nucleotide synthesis, you said?
Sequence.
Nucleotide sequence. Okay.
Those which
Nucleotides. Basically, biochemical methods focus on something like nucleotides. Okay, right. So all those things. So biochemicals in our body and methods to understand them, methods to monitor them, methods to detect them. So all these things should come under biochemical methods. Okay, right. Now let me define it a bit more specifically here. So biochemical genetics refers to study of genetics in terms of the biochemical events. Okay, so it is the area biochemical genetics. Biochemical genetics is an area. Biochemical genetics is an area, okay, that deals with the study of biochemicals. Deals with study of biochemicals. Study of biochemicals. Okay, right. Such as so sorry. What type of examples are these? Such as, so these are the list of things. This is to explain you the scope. You don't have to write all of them, but things related to these processes I mentioned here, some of them you already know. So we are focusing on that part of genetics. Okay, connected to what, or in connection with what we are learning here. DNA replication. So DNA, formation of DNA, synthesis of DNA, how is DNA made in our body, and replication of DNA. Genes, how genes are expressed, right? You all know what is gene expression, am I right? What is gene expression? Do genes actually express?
Showing a particular phenotype or a genotype.
Correct. So basically transcription, translation. From DNA sequence, from genes, formation of mRNA, formation of protein, that process is called gene expression. That process is called gene expression. So how genes get expressed, how mRNA is made, how proteins are made, they come under biochemical genetics. And proteins, we know that many enzymes are proteins, correct? So synthesis of those enzymes, how are these enzymes made? How are these proteins made? Because enzymes are the key players of biochemistry of our body. Many biochemical reactions in our body take place because of enzymes. So how are they made? How are they synthesized? Are there anything related to their abnormalities are studied under biochemical genetics? DNA replication, gene expression, synthesis of specific enzymes. Okay, and metabolic pathways, metabolic reactions, meaning chemical reactions, that biochemical reactions that happen in our body, like how DNA is made, how proteins are made, how ATP is made. So they're also part of biochemical genetics. Okay, so they're also being discussed as part of biochemical genetics. Okay, and synthesis of essential components, amino acids, nucleotides. So as I already telling you about this. So these are also part of biochemical genetics. Okay, and also finally structure and function of biomolecules. So what are biomolecules? Biomolecules are these things: proteins, carbohydrates, lipids. These are all called biomolecules. You will study this in NCERT textbooks. Biomolecules, there's a dedicated chapter on them. Okay, so these type of things. Okay, so this is what biochemistry is. So we are studying about various biochemicals and the processes related to them, starting from synthesis, their regulation, okay, their function. All these things come under biochemistry. So what do these biochemicals include? They include anything from amino acids, nucleotides, and the products that they make. Nucleotides make up DNA, amino acids make up proteins. We study about them, like replication of them, synthesis of proteins, and gene expression. And some proteins act like enzymes, so synthesis of specific enzymes, and all sorts of metabolic pathways. Like metabolic pathways, something that you all know very well, like something like digestion. When we digest the food, and the respiration process, converting digested food like glucose into energy. So ATP synthesis by utilizing sugar rich, like sugars like a glucose, that is also a type of metabolic reaction. All various metabolic reactions that happen inside our body, there must be genes coding for these specific type of proteins and enzymes which are dealing with them. So because all the characteristics that we talk about either directly or indirectly defined by our genetic information, our genes, our DNA. Correct, but DNA directly doesn't work, so it has to work in the form of proteins. So all those components that we study at the molecular level related to this DNA, proteins, enzymes, and their constituents is called biochemical genetics. Okay, are you getting an idea about what is biochemical genetics? Okay, I'm going to give you actually brief summary on this as well in the handout that is given to you. So I also briefly explained. If at all, if I have to add anything to that, like hormones, even hormones, some hormones, not all hormones are proteins, okay, but they're also biochemicals. In biochemistry, we specifically study about hormones. How are they made? What type of functions they have? Like, for example, if you know hormones like oxytocin, someone was mentioning, growth hormone, oxytocin, growth hormone, and we have insulin, glucagon. So like that, there are several hormones produced by various glands in our body. It also comes under biochemistry, and biochemical genetics is a genetic component that deals with this. Okay, so like that, we understand about the various components like genes, proteins, metabolism, they all come under this. So this is a brief idea about what is the scope. So you can write methods related to them. There's so many methods. Even you probably find books of 1,000 pages, 1,200 pages, just explaining about these concepts and methods. But for us, briefly, if we have to write something like a short note, 10 marker question or a 15 marker question. So would you be able to explain what is biochemical genetics? Would you be able to explain what comes under it? A case study, one or two methods for it. Okay, that should be the scope of this class. So biochemistry deals with all these things. As I was telling you already, RNA, DNA, those components. There are many techniques that are there. The techniques that are used to quantify DNA, RNA, checking how much of DNA, RNA you have in a given sample, how much of sugar. For diabetes, normal biochemical test that we do. In the blood, we measure sugar levels, glucose levels we do. That is also biochemical method. Even detecting presence of antigens, antibodies that we're talking today in COVID-19, this could be biochemical method, but also part of immunological method. If you're confused, let's say we also have next topic is immunological methods. So there is overlap between biochemicals and immunological methods. Those biochemicals which take part in immunology, immune system activity, are also immunochemicals. So bioimmuno can also have some overlap. Okay, like antigens, antibodies, if we talk about that, they are also biochemicals, but they come under immunologicals. Okay, right, bit of history, like where did this biochemical genetics part had started? Okay, so if you see any standard textbooks, they discuss about this concept. So I wanted to briefly explain you this. So actually, our understanding about biochemistry had started much earlier than that, but a stronger foundation in this biochemical part had come in the beginning of 20th century. Uh, so a couple of scientists are actually gets credit for this. One being the Archibald Garrard. Archibald Garrard, he's a physician from UK. He was a doctor to the royal family, Archibald Garrard. So he is well known for his studies on this. And especially from his work, he pointed out on something called inborn errors of metabolism. Because think about it, if everything goes all right, I don't think you need to worry too much about this. In methods, most of the times we use this technology for monitoring any problems or understanding any issues with biochemistry of the body. One such problem is errors in metabolism. Some of the metabolic reactions are biochemical reactions. There are some defects that when we are born with defects in those processes. So these are called inborn errors of metabolism. I'll show you one or two examples on this. They're called inborn errors of metabolism, inherited diseases, which results in inability to make certain enzymes are the common examples. So what am I trying to tell you here? When we are born, some of us actually show problems in metabolic reactions. Okay, So most of the times these problems are because of maybe a gene is mutated or affected whose product is responsible for that reaction to happen. Okay, wherever you are not able to follow, do stop me. So here we're talking about enzymes. I'm going to give examples on this. So enzymes which actually control certain metabolic reactions in our body. And gene responsible for that enzyme, gene responsible for producing or coding for that enzyme, if it has a problem, then what happens? We will see issue related to that. What do I mean by that? I'm going to give an example, but before that, let me give you a brief overview on this concept. So, study by this Archibald Gerald on the inborn errors or birth errors that we have from the birth suggested that genes dictate phenotype through enzymes that catalyze specific reactions. So, meaning they thought that there must be some connection to enzymes. So, they thought for every phenotype there may be enzyme. So, every time we have a characteristic, they thought there must be an enzyme involved in it. This is how they initially thought, because remember, we are talking about an era right after Mendel. So Mendel could only give us idea about the inheritance pattern. But later they found out the pieces that result in inheritance, like genes, DNA structure, those they found out. So we're talking about early 1900s. So it's a time when they started thinking, okay, we see the connection between something coming from our parents to us. So they thought, okay, we have DNA sequences. What are they doing? They may be producing enzymes. Those enzymes are responsible for our characteristics. So they were trying to make connections here. They thought initially, okay, a gene gives rise to enzyme gives rise to character. You can put like this: gene, arrow mark, enzyme, arrow mark, character. Okay. So later, this is one concept, one gene, one enzyme concept, mostly generated from the observation of errors in metabolism. Errors in metabolism. Errors in metabolism, where enzymes are basically cause this problem. Okay, so later even George Beadle and Edward Tatum. So these are the two other scientists, George Beadle, again from 1903 to 89, and Edward Tatum. So these are also two who investigated these pathways, metabolic pathways, how reactions happen, where do they start, how many products they have. They studied these type of things by working on a fungi. Okay, so these people have proposed this concept I was just talking to you, like one gene, one enzyme hypothesis they gave. They said a single gene specifies synthesis of an enzyme. An enzyme is responsible for certain characteristics in the body. They also received Nobel Prize for this concept. So Archibald Gerard and Beadle and Tatum. Archibald Gerard worked on metabolic errors, and Beadle and Tatum understood that missing of metabolites would also affects a function of one particular compound. So you don't need to know this, but just conceptually look at the idea how they came up with these things. So the Beadle and Tatum, what they have done to prove this, they worked on a fungi. So what they have done, they actually took fungus and put some X-rays on them, shed some X-rays on them. So they expose them to X-rays. And don't write anything on this. This is just to develop the idea. Okay, don't have to write in the exam. I don't think we need to get into these details. So they took fungi and they shed some X-rays on them. Fungus normally multiplies in the form of spores. They have tiny structures called spores, right? So those spores they have taken and they distributed them into several tubes, and in each tube they had minimal medium, we call, or food that is required for them to survive. Okay, so food that is required them to survive. But because of exposure to X-rays, what happened? Some mutation happened in them. Some of them didn't grow because some of them are they unable to produce some missing compounds. So what happened here, when they got exposed to X-ray, some maybe a gene or two got mutated. and the product of that gene is missing now, is defective now. So they could not synthesize something new. So if you provide them a nutritional, like a, we call it nutritional mutant, or you provide them the food or nutrition you provide to them, some of them could not grow. What happened? That mutation affected its ability to consume the food. The mutation affected its ability to consume the food. So through these studies, they came up with the idea of one gene, one enzyme. So there must be a gene that was affected because of X-ray, and because this gene function was missing, it could not digest the food. It could not utilize the food that is given, and it was not surviving. So through this type of specific studies, they've actually come up with the idea of this one gene, one enzyme concept. So overall, what are you seeing here? Either we're talking about errors in metabolism or knowing the connection between some small metabolites and their role in our body's functioning. They found out that definitely biochemistry has a big role. Knowing those biochemicals, metabolites is very, very important. This is what they figured out through these studies. So this is where I think where biochemical genetics role field has started. But of course, we are more interested on not just these concepts, we are more interested on the methods part. Are there any examples that we can write in the exam? Correct? So in related to this inborn errors of metabolism, so the particular disease concept that if you see, so as per the Gerard, he thought if we have like a substrate called A, the substrate has to be converted into a product called B, and B finally has to be converted into C. So A should be converted into B, B should be converted into C. Like that, if you take the glucose, glucose gets converted into some other compound, like glucose 6-phosphate we normally say. Like that, glucose and other products will form in the body. It's like chain reaction, one after other, one after other. Couple of things happen, finally giving rise to the product. So glucose finally gives rise to ATPs. Like that, A should get converted into B, B should get converted into C. So this is a normal procedure. Okay, but if things go wrong, in the previous example that we have seen in fungi, if they are exposed to X-ray, some of these genes, especially whose products are responsible for converting A into B or B into C. So let's say mutation has happened in them, and we are missing now. This organism is missing enzyme responsible for converting B into C. So what happens in those cases? If a gene responsible for producing an enzyme responsible for converting B into C, what would happen to that reaction? Would that reaction continue? Would that reaction happen? If a compound required for converting B into C is absent, what happens to it? What would be the outcome?
C will not will be get produced.
So C, production of C will not be there. So whatever job of the C is, that will be missing. Very good. What else can happen? So absence of end product. Blocking a metabolic pathway like this will lead to absence of end product. So this product is not being made. At the same time, what happens to B?
Mutation to be give rise to new product.
So basically, B may be able, B accumulates. B will remain in the body. It will accumulate. Either B accumulates in the form of B, or B may get converted into something that is more toxic. So to keep it simple, you can even forget about D. You just show this scenario: A to B, B to C. So whatever we need, an enzyme needed for converting B into C is missing because of a mutation in a gene. So what happened? Because of this, C will be absent in the body and B will accumulate. You put an arrow mark for B, increased arrow mark, like excess of B may be accumulated in the body. Don't even write about D. So B, accumulation of B may also be toxic to us. So these two possibilities are there. One, the product will be missing. In this case, C will be missing. Two, B gets accumulated and B may be toxic to our body. Okay, there are many such problems are there. At least over 550 such problems are there. So 550 diseases, inborn errors of metabolisms are there. Over 500. Remember, at least over 500. So definitely a common problem. So very important for us to understand about biochemical genetics. Okay, this is a general idea. Now I'll show an example for this. That's why I wanted to write briefly. A goes to B goes to C. either B gets accumulated where C will be absent in if error happens in this type of reaction. Okay. We don't need this. One example. Example 1, alkaptonuria. First example is alkaptonuria. I'll give you 2 examples. You write whatever you are comfortable with. So write down the name of the disease, alkaptonuria. Excuse me. So the name of the disease that we are learning here is alkaptonuria. What is alkaptonuria and what causes this? So it's an inborn error of metabolism. It's also a genetic disease. It's an autosomal recessive disorder. We talked about in pedigrees. So its inheritance pattern is autosomal recessive. So not present on sex chromosomes, present on one of the autosomes, and recessive, meaning 2 defective copies are needed for causing the problem here. Okay. Okay, right. So in this inborn error of metabolism, what happens? You see on the flow chart here. So normally what happens? We have amino acid called phenylalanine in our body. Phenylalanine. So when we eat the food, many foods that we eat provide us phenylalanine. So this is one of the building blocks of protein. It's an amino acid, which is a building block of protein. So what happens is phenylalanine should get converted into tyrosine, another amino acid, and then like that, multiples of them. So instead of writing A, B, C, D, in the previous image we have seen A, B, C, D. I'm telling you the example. So in this case, A is phenylalanine, and then A gets converted into tyrosine, and then something called hydroxyphenyl pyruvate. So won't be able to remember this, right? So just say that it is something related to the phenylalanine metabolism. So in the process, the fourth one, this is what is important for us, is homogentisic acid. So in this reaction, okay, where phenylalanine metabolism it is called. So phenylalanine is an amino acid and it gets processed in the body. So it gives rise to, at one point, it should give rise to homogentisic acid. In some people, what happens because of this alkaptonuria, because of a mutation, okay, in them, what happens? This homogentisic acid, the enzyme responsible for converting homogentisic acid to the next product. Okay, so this is a type of acetoacetic acid. So this one, the enzyme is absent here. The enzyme to convert homogenistic acid to the next compound, like in this case A, B, C, D, E, D to E is absent because of a defect, because of a mutation. Then what happens to homogentisic acid? As we have seen in the previous generic picture. So one, the acetoacetic acid, that mallyl acetoacetic acid will be absent. Plus, what happens to homogentisic acid?
It's accumulated.
It gets accumulated. It gets accumulated. This is a key idea you have to get. You don't have to by heart the names, just mention as part of phenylalanine metabolic reaction. You put couple of arrows. Phenylalanine, put 1, 2, 3 arrows. After 3 arrows, you write homogentisic acid. And put another arrow here and say that enzyme responsible for converting homogentisic acid to the next product is affected. So gene coding for that enzyme is affected. Excuse me. Right. So when that happens, so this this gets toxic to the body. The homogenesic acid accumulation is found in urine, plasma, part of the bones, connective tissues. You will find them in various tissues in our body. So this homogenesic acid starting to getting accumulated. Okay, it starts to getting accumulated.
Sir, type 2 sugar example.
Sorry, what?
Type 2 sugar.
Type 2 sugar can be an example for, it's not inborn error of metabolism. Okay, in the diabetes can be taken an example for this type of metabolic disorders for sure. You can take it as an example for that. There you can mention, because there it is not a genetic problem. That is why it has a genetic component to that, but it is not called a genetic problem. Whereas these are genetic problems. So, but there also is not genetic problem because of the defects in metabolic reactions does diabetes take. So you can definitely mention, but standard in textbooks they talk about these examples. But definitely diabetes, type 2 diabetes can be as an example for this. Accumulation of glucose in the body, which becomes toxic to liver, heart, kidney. That can also be mentioned. But there the problem is mostly because of the insulin, which is also a hormone biochemical product. Definitely you can write diabetes as an example for this as well. But more standard examples in the textbooks are these. Okay, right. So accumulation of homogenesic acid would result in the couple of problems. One being the urine becomes dark. As you see urine here, from left to the right, dark urine. These people actually produce dark urine because mainly this homogenesic acid, when it undergoes oxidation reaction, oxidation is one common chemical reaction, it becomes dark. So the dark coloration in the urine is because of presence of this homogenesic acid, a product of homogenesic acid. So people may even have dark ears. You see here, her ear got darkened. Some people have dark noses because of accumulation of this dark color product. In the eyes also, people may have such a product. Okay, even in the eyes, joints. So these type of problems are normally seen. Okay, so this is one very good example for inborn errors of metabolism. So see how much you can remember and write here. Okay, you can say phenylalanine, couple of arrow marks, homogenesic acid, and a defect in this part, in this portion when in this part when homogenesic acid needs to be converted into something else, but thereby resulting in accumulation of homogenesic acid. And where does it accumulate? Various parts of the body, urine, plasma, cartilage related to the bones, and also ears, nose, okay, eyes. So various tissues have this with a darkened color. It is not deadly, though it doesn't cause a death in humans. A rare disease, but is the manageable disease, not too much of a problem. But how do you measure this? How do you know whether someone is suffering from alkaptonuria? So change in color of the urine and standing to. So if you collect urine and leave the urine, so urine will turn brown to dark like this. So simple biochemical test, because when this urine containing homogenesic acid is exposed to oxygen, it gets converted into this. Okay, patient with alkaptonuria, as you see here, it becomes dark. So this urine test is a positive test for. Okay, this is a one test. I think you can mention that. There are also other biochemical methods. Don't worry about them. This is adding ferric chloride, silver nitrate. So they're chemical methods. But this is already, if you mention it, when you actually collect the urine and reacts with oxygen, it will become dark. It gets oxidized. So people, because of this, may have certain problems. So because it gets accumulated in the ears, hearing loss may be seen. Some people, heart defects are seen. And kidney stones, bone fractures, these are also seen in people with alkaptonuria. Of course, I was telling you it is very, very rare. 1 in 250,000 people, 250,000 people, population-wise variation happen, but rare problem. So most of the times for these diseases, avoiding that protein, phenylalanine is a protein, right? Avoid foods rich in phenylalanine is the best management method for this. Avoiding the phenylalanine rich foods.
Sir, the benzene that we have, the same structure must be for phenylalanine, right?
Benzene is, yes, structure looks like benzene structure, but it has more complex structure to phenylalanine.
So it is as such not poisonous to normal people.
No. Phenylalanine is a common compound inside our body, so it's not poisonous to us. No. You're not asking about benzene, right? You're asking about phenylalanine. Phenylalanine shares some structural similarity with the benzene. Okay, it's a phenyl group. So normally to the benzene, we have a functional group called phenyl group is added to that. Okay. So it is not toxic, but it has more complex structure than the just benzene. Benzene, on top of that, you will have a functional group attached to it. Okay. Right. So second example I like to give you here is phenylketonuria. Concept is similar. So we just follow what we talked about. The disease name is phenylketonuria. Previous one was alkaptonuria. This is called phenylketonuria. So yes, you see that this is the structure I think you're asking about. So phenylalanine is a chemical structure. So this is functional group attached to that. So this is in a way connected to the previous example, but here the problem is in the first step itself, phenylalanine with phenylalanine itself. Okay, I was telling you phenylalanine rich foods I was talking about, and I was telling it's an amino acid. So the foods like fish, pork, cheese, breast milk, they all contain phenylalanine. Fish, pork, cheese, breast milk are some examples of phenylalanine rich foods. Okay. So just write so much. You don't have to write everything. Just a brief example that you have to write here. Phenylketonuria, also a genetic disorder where phenylalanine accumulation takes place. Phenylalanine accumulation takes place. I'll show you this in the image here. So this is phenylalanine. Phenylalanine will become tyrosine. Remember from the previous structure that we have seen this? Phenylalanine should become tyrosine, right? So at this step, there is a problem because we need an enzyme called phenylalanine hydroxylase. Okay, or you mention the enzyme if you can remember. If you don't remember, just say that in phenylalanine metabolism, so it becomes tyrosine with the help of an enzyme. This enzyme name is phenylalanine hydroxylase. A defect in a gene coding for this. Defect in a gene coding for this. So what happens because of that? We'll have phenylalanine accumulation takes place, and that disease is called phenylketonuria. So where is the problem here? Problem is in this enzyme, which is responsible for converting phenylalanine into tyrosine. So accumulation of phenylalanine takes place in the body. Okay, right. So what is it? Is it actually toxic to us? It can get toxic. So normally, if it is in certain concentration, within the limits of certain concentration, it doesn't cause problem. Very much like glucose. Glucose in a normal concentration don't cause a problem. I think it has to be above 0.1% in the blood. If it is above 0.1% in the blood, so we see negative effects of glucose. Similarly, even phenylalanine, if it starts to accumulate in the body to high concentrations, we start having the problem here. So they don't, babies look normal. You cannot find out much of a difference or much of a phenotype in them. So, but the problems come if they don't get treated, if they don't get diagnosed, they don't get treated, problems show up. Like for example, people may have learning disabilities, behavioral problems. Okay, so these type of components are epilepsy. So these type of problems can be seen in them. So what happens if phenylalanine gets accumulated in their body? So some problems eventually show up, but by birth they may not see too much of an issue here, too much of a problem here.
Sir, what is epilepsy?
So epilepsy is a nervous system problem. It's a nervous system related disorder, a nervous system related disease it is called. Okay. So as I was telling you, sometimes people actually have some sort of disturbance. They may experience something like fits or convulsions are there. So they may faint. They may actually have convulsions or fits may be seen. So that disease is called a nervous system disorder. Is called epilepsy. So it leads to fits, leads to fainting in people. Okay, there are multiple reasons, and here one reason is in this case it is metabolic disorder. So usually we see that fits and these comes because of the abnormal electrical activity. Suddenly you have some amount of extra chemicals being produced. So because of signaling issue, what happens? Electrical activity. It's like power going on and off to the parts of the body. That is why they have that quick movements or jerks in muscles. Okay, so these are some of the problems here with phenylketonuria. So something with the other, same as alkaptonuria, the best solution here is to manage the food. Phenylalanine rich foods, as I was telling you, meats, several meats, fish, even eggs, they say, dairy products. Okay, they are phenylalanine rich. So managing them, meaning avoiding them as much as possible. So eating specific type of protein foods where phenylalanine is less. Okay, so keeping an eye on the diet, especially the protein diet that they take, is the best way of managing the problem. It doesn't kill people, as I was telling you, but they have to realize it enough, early enough, and then starting to manage the problem by avoiding the foods that causes the accumulation of phenylalanine. I think we talked about this. These are all examples we talked about them in the 9.2. Conditional lethal. Conditional lethal. Only when they get accumulated, they cause problems. So we can avoid them by choosing the food. Okay, right. So these are the two examples. These are examples for biochemical genetics part, and especially inborn errors of metabolism. Inborn errors of metabolism. Okay. Next, we'll move on to how do we study them? What is the best way? What type of biochemical methods are required for studying them? Let's understand that. Okay, I'll give you two such examples of methods here, because as the name says, biochemical methods. Let's also focus a bit on the methods part of it. So far, I explained you what does it mean by biochemical genetics and what areas does it come under, what areas come under biochemical genetics part. And we discussed about the brief history of biochemical genetics with examples. So inborn errors of metabolism, one gene, one enzyme concept. Later it went on one gene, one polypeptide. And then today we know that one gene can give rise to a polypeptide, may not be even protein. Okay, right. So basically, we have definitely lot of things to study under biochemical genetics. But good example is to talk about inborn errors of metabolism. Okay, example, you give one of this phenyl ketonuria or alkaptonuria, you give it as an example. Next, we're talking about methods. What type of methods are useful to study these type of products? Okay, so most commonly we hear about electrophoresis. Electrophoresis is a method. Okay, one method that is routinely used for biochemistry. There are more simpler methods than electrophoresis as well. Like I was already telling you, diagnosing presence of homogentic acid in urine tests are also biochemical tests. So there are ways to identify the chemicals that are accumulating in the body, like glucose, sugar tests. For diabetes tests, diabetics they normally throw a test to check for the amount of glucose in the urine. Sorry. So those tests and urine tests for these type of things, some of the blood tests, they can be considered as biochemical tests. But bit more sophisticated technology we'll talk about here. This is called electrophoresis. Write down the name of this method. So subheading should be methods. Excuse me. So electrophoresis. I'll give you a brief example, a brief conceptual explanation I'll give you. So what is electrophoresis? So electrophoresis is a method where biomolecules, like, listen to me first and then you write, okay? Because when you write as a summary of what you understood, you don't have to refer to anything, otherwise it gets complicated for you. So what is first let's understand the concept. So it is a method that is used in analyzing biomolecules or metabolites. Because we're talking about biochemistry is dealing with metabolic reactions and the components related to that. So here, DNA, RNA, proteins are metabolites, correct? They're also biomolecules and metabolites. So studying them, we need tools or methods to study them. They can be discussed as part of the biochemical methods. Things to study: DNA, RNA, proteins. And this electrophoresis is suitable for all three of them, for DNA, RNA, and protein. So common method, electrophoresis is a routine method used in analyzing, write down the statement. So electrophoresis is a routine method used in analyzing DNA, RNA, RNA, proteins, etc. So concept wise, you know what? When you are learning about DNA, RNA, proteins, one question you always ask is their size. How big are they? Are we looking into a small RNA, a small DNA, or a big DNA, or a big protein? So size is a very good measurement. Okay, and electrophoresis can differentiate these molecules based on size. So what do I mean by that? Let's say inside our body, you want to see if we have a problem with one particular metabolite. Let's say a problem with a protein. What do? You collect body sample, you collect blood, you collect blood plasma, okay, and look for presence of that protein. So one criteria to detect it is its size. So if you're able to differentiate the size of that protein from the other relevant products, then you can, can actually can be measured. So based on the size, separation needs to be done. Electrophoresis is a method, okay, that separates biomolecules. So next point, if you want to write, electrophoresis method separates the above mentioned biomolecules. We talked about the biomolecules before, right? The above biomolecules like DNA, RNA, and proteins based on their size. Based on their size. Based on their size and charge, also on charge. But charge we can actually fix. Charge we can fix, but size we don't want to fix. Size we want to identify. So what do? So I'm now coming to the method part. So far I told you a brief idea about electrophoresis, where is it useful. Methods wise, you see some sort of a gel-like structure. Gel is like semi-solid, right? Semi-solid like structure. So you take gel-like material and normally we punch holes into it. So gel-like material is taken, we punch holes into that. So why do you punch holes? Let's say we collected your blood sample or plasma sample of a patient. So you actually place that into that hole. So you take a piece, okay, with the gel-like material, and place the sample in the hole. You place the sample in the hole. When you place a sample in a hole, what happens? You put it under the influence of electric current. So you place this under electric current. So engineers, I'm sure you all know this, you understand this. We have two electrodes, right? Positive electrode and negative electrode. So in a process called electrolysis, what you normally hear. So if you actually put something like this, depending on the molecule may have certain charge. If you take DNA or if you take proteins, DNA has a negative charge. DNA usually have a negative charge, and proteins have mixed charges. Some proteins have positive charge, some proteins have negative charge, some are neutral. So molecules may have some charge. So in electric field like that, let's say that we are working with DNA. So if DNA has a negative charge, what is the best thing? You place the DNA on the electrode that is negatively charged. Okay, and when you provide electricity to that, when you actually push these molecules with the electricity, then what happens? They move out to the opposite side because negatively charged DNA gets attracted to the positively charged electrode. Negatively charged DNA gets attracted to the positively charged electrode. But we have DNA, maybe if you're talking about DNA, we have multiple molecules of DNA. Or if you're talking about proteins, we have mixture of proteins in the body. You are interested in identifying a specific protein, but there are mixtures of proteins. So what do? Under the influence of electricity, what happens? Because of the negative charge, assume that even protein has a negative charge. Assume like that. So for protein, we can fix it. Okay, it's a different method, but somehow we make protein to have uniform charge, uniform negative charge. But we have mixtures of protein. So what do? In the electric field, what happens? It moves towards the positive electrode. In the process, size-based separation takes place. What do I mean by that? I'll give an analogy like this. Let's say you are running a race. You are running a race for people with different weights. Assume that someone 50 kgs, someone 70 kgs, someone 100 kgs. Who would run fast? 50, 70, 100? Generally, theoretical question. So you are moving them, you are asking them to run from one side to other side. Who would run faster? 50 kgs, 70 kgs, 100 kgs?
50.
50. Okay. Can 100 kgs person run faster? No, because of negative correlation. How much body weight they have to carry on them? They have to carry on so much of body weight to run, right? So maybe 70 kgs person also can run faster. But just I'm trying to tell you here. So there is a correlation between the weight of the molecule and how fast they move. So what happens in these cases? The proteins in the electric, under the influence of electricity, they actually move towards the opposite electrode. In the process, they get separated based on the size. So which one will move faster? If you see on the image here, so they added sample here. Okay, and in this particular case, if you are allowing them to move towards the opposite electrode, in this case, positive electrode from negative side of the electrode. So the bottom one, let's say there are many of them. Out of the examples I was telling you, 50, 70, 100, which one will be at the bottom? The one with 100 units weight or the one with 50 units weight will be at the bottom here or away from the starting point. In this case, which one will be away from the starting point? Let's say take here. So this is starting point. We have, is the largest one will be here, heaviest one will be here, or the lightest one will be here, away from the starting point.
Lightest one.
Lightest one.
Lightest one. Why? Because lightest one can run faster or can move faster. So the one with 50 will be here, the one with 70 will be here, the one with 100 will be here. Like that, size-based separation takes place. That is what is called electrophoresis. Okay, could you understand this? Could you follow this? Briefly know this. I've also given you in the handout. I've briefly explained this process. I've given a bit more details, but this is what the concept here. So electrophoresis is used in separation of nucleic acids, also for proteins, also for other metabolites. And based on what? They separate based on their charge and size. In this case, molecules separate based on their charge and size under the influence of electric field. I myself have summarized this in the handout. Under the influence of electric field. So what happens here, where negatively charged molecules migrate towards the positive electrode. Positive electrode is anode. You can remember, right? Anode, ANOD, anode, or positive electrode. So this is actually I have given, I'm just explaining it, and the handout I'm going to share with you after the class. Read it, read the electrophoresis part in this. You will be able to understand this. Or for now, if you want to write like a key points, like a flow chart or just points. Okay, and the migration is determined by the size of them. Migration is determined by the size of them. Now their movement is defined by their size. Lighter ones move faster, heavier ones move slower. Lighter ones move faster, heavier ones move slower. So this is a theme. Like that, size-based separation can be done. But how do you know which one is a protein that you are interested in? Let's say you're looking for an abnormal protein. How do you know where that is? So they have to run a reference for it. So basically, what do? We call it ladder, but a size-based. So a product whose fragments and their size is known. So like a reference. So when they compare these products with the reference, the size of the product of your interest can be identified. Or based on size, you can separate or you can identify the product of your choice. So what do they do here? Meaning, basically, let's say that this is a reference point. So you know each fragment. If there are, to make it simple, let's say there are 4 sizes, only 4 fragments in the simplified image. Let's imagine that. So the bottom one is 200 units, or 200 base pairs if it is DNA. If it is proteins, 200 amino acids, normally say, because they're measured by amino acids. So let's say bottom one is 200. The one in the center is 100. Then 50. Then 25. Like that, 25, 50, 100, 200. Imagine there are 4 fragments, and you know with this product that the bottommost one will be the smallest, that is 25. The second upper one will be the 50, third one will be the 75, and then the 100. I think I told you reverse in the before, right? So smallest one will be, lightest one will be 25, then 50, then 75, then 100. Let's say you have something like this, like a ladder, like a reference point for you. Would you be able to identify something close to this? So this is, if this is 20, if this is 75, let's say this is a protein that you are interested in. Would you know this or not? Because it is close to 75. It is between 75 and 50 units fragments. So from that, from comparison, you will know the size of the protein that you are looking for is 50. Then you know this is what it is. And there are many methods where you can quantify its presence. Is it too much or too little? What should be the normal range? If you see your blood tests or other reports, you will see the normal range for it, right? Similarly, we can identify the range for this substance, if it is too much or too little present in the body. So like that, electrophoresis technique is routinely used for analyzing proteins and other metabolites. Okay. Right. So if someone is having a problem or a difficulty with some proteins in their blood, okay, in their body, so these type of methods can be used to identify their presence in urine samples or in blood samples. So one can detect the abnormal proteins or abnormal levels of proteins, if not necessarily the abnormal type of protein. I'm going to tell you in the next example abnormal type of protein with a case study. But here abnormal levels of proteins can also be detected. Okay, so problems or molecules found in urine blood samples can be detected using such methods. So basically what are we detecting here? We're detecting biochemicals, right? Okay. Right. So next we'll take one example of a biochemical problem. We can take metabolic disorders that you mentioned, we talked about already. So like we can talk about glucose in diabetes as an example. One such example I'd like to give you here is hemoglobinopathies. Write down, it's like a case study of a disease. Okay, in biochemical genetics, let's learn how variation in one particular biomolecule, like a protein, can be studied using biochemical methods. Here, disease that we are focusing is hemoglobinopathies. The name of a disease is slightly complicated. Hemoglobinopathies, it is called. Hemoglobinopathies. As the name suggests, it is a disorder related to what? Hemoglobin in our body. So you all know what is hemoglobin. What does hemoglobin do?
Carry oxygen.
They carry oxygen. They supply oxygen. They are transporters of oxygen. So hemoglobin is responsible for carrying oxygen. Correct? So this is the basic function of it. So is it very important to carry oxygen in the body? It is definitely an essential function. So it's an essential protein for us to survive. So we routinely see that either we produce abnormal hemoglobin molecules, either we produce abnormal levels of hemoglobin molecules in our body. So hemoglobin is a transporter of oxygen, which is essential for our survival, and which shows some problems. So sometimes we have abnormal types of hemoglobin produced. HBS, HBC are two examples. We talked about HBS already. Any idea what is hemoglobin S? Lethal genes we talked about. I think at least two instances we discussed about this disease. HBS, hemoglobin, mutation in hemoglobin results in change in shape of RBC.
Sickle cell.
Sickle cell
Sickle
anemia.
cell anemia. Very good. Sickle cell anemia. HBS is the sickled character. So it is because of the mutation in the one of the hemoglobin chains. You write this example. This is enough. But otherwise there are many. So there are so many different types of them. HBS, HBC. HBC is a particular type of mutation. So HBS normally what happens? Glutamate is replaced by valine. Both of them are amino acids. Instead of glutamate, valine is present. In case of HBC, instead of glutamate, lysine is present. So same position but different substitution. Different amino acid is present because of the mutation. One results in the sickle cell disease, another one results in the hemoglobin C disease. Both of them can result in anemic condition, especially sickle cell disease can result in the abnormal flow of oxygen in the body, and it is an autosomal recessive disorder. So what happens, and because of that, so in a homozygous recessive condition, people will die. Okay, I think we were talking about that in the last class also, if I'm not wrong. Pedigrees, I think I mentioned in the pedigrees, right? Sickle cells can gets basically they can get accumulated in the blood vessels, thereby blocking the blood vessels because they are not circular anymore. They don't get easily squeezed through the blood vessels. They're like crescent. They actually get stuck and stop the blood flow. So like that, sickle cell anemia or hemoglobin C disease, both of them can result in some form of anemia, anemic conditions in them. So these type of abnormal alleles can be present in us, or sometimes abnormal quantities can be seen. So one very common disease called thalassemia. You probably heard about thalassemia. Thalassemia is a very common problem in India, thalassemia. And here again, because of a mutation, we produce lesser hemoglobin. Our defective hemoglobin is made. So overall hemoglobin production is less. So thalassemia also is a type of anemia. Anemia means loss of blood or lack of enough blood, correct? Anemic condition we say. So like that, there are many defects in hemoglobin. There are many defects in hemoglobin. Sickle cell anemia, thalassemia, HBC disease are examples.
Sir,
So-
sickle cell is so sickle cell trait, जो carrier condition, is that recessive, heterozygous recessive or heterozygous dominant?
So is it dominant or recessive you're asking, am I right? So heterozygous means having two different alleles. Only if it is a dominant allele, it would express. But this we normally call sickle cell trait is where you have one mutant allele, one normal allele. Given that this condition is recessive condition, so heterozygous condition, it won't express. So we'll not have problem because actually what happens is here in sickle cell anemia, there are few RBCs with abnormal shape, they get destroyed. But other allele is producing normal copy of RBCs. So we will manage with that RBC. But when both the mutant alleles, both HBS come together, this is homozygous recessive condition where problem happens. Okay, you don't call anything like heterozygous dominant, but you normally say that dominant allele being dominant in the heterozygous condition. But it's recessive allele, so it requires homozygous condition for showing the problem. How do you study these type of diseases? If the disease is there must be a method to actually study this. So how do you study abnormal hemoglobins in our body? So we definitely need a method. Electrophoresis we talked about. Let's use that electrophoresis method for studying this problem. So we already discussed about electrophoresis. How does electrophoresis work? Correct. Now let's see how is electrophoresis used in studying hemoglobinopathies or diseases related to hemoglobin. Okay. So as we already talked about, based on the principle of electrophoresis, it should separate various types of hemoglobin provided they have different sizes. Correct? If hemoglobins happen to have different sizes, they can be separated using electrophoresis and identify their presence. And also hemoglobins have different charges as well. They also have different charge. So the way they get attracted to the electrode is also different. Okay, so I'll show you an image for this. So they have picked up from a research paper. So what are you basically learning? They're talking about hemoglobin electrophoresis. They put several different types of hemoglobins taken from different patients here. So there are different varieties of hemoglobin that you normally see. Like for example, here this is a normal condition. People with no hemoglobin related disorders, their pattern should be like this. We have different forms of hemoglobin, A form, F form, A2 form. So don't have to worry about the forms here. Just see that this is a normal healthy pattern. Normal healthy pattern for hemoglobin looks like this. But a person with beta thalassemia, major or beta thalassemia trait, so meaning heterozygous condition, homozygous condition, what do you notice being different from this normal condition and beta thalassemia trait or beta thalassemia major? Or you pick one of them, doesn't matter. So what difference do you see between these, mainly with reference to this? What difference do you see in them, in both of these?
Reducing A and increasing F.
Correct. The size of this band is reduced, meaning its quantity is less. So production of A type of hemoglobin chain is less. F chain, is there any effect on F chain? Slight shift in the size, but I don't know how much label is that from this image. But A2, increase in A2, decrease in A1 or A, increase in A2 levels of hemoglobin happen. So some form of difference you can easily recognize here, or you can make out here.
So in a single blood we have different hemoglobin type.
Yes, correct. I think we're going to talk about that in the genetic markers. One unit we're going to discuss that. In our blood, we have different chains of hemoglobin, and even with age, you see variation in hemoglobin types in our body. That is true. Okay, right. So if we have a pattern that is like this, which can be differentiated using electrophoresis, electrophoresis is a great tool because it is not too complex. Normally, we can run these type of experiments in half an hour, 45 minutes. So just we need to figure out what needs to be added for that. But with electrophoresis technique, you can differentiate between normal people and people with abnormal levels and abnormal types of hemoglobin because they have a tendency to show like this variation in their protein levels can be seen here. Like that, in this particular research paper, as part of hemoglobinopathies, they have established a method using electrophoresis, which is a very simple method and a quick method to differentiate normal condition and abnormal hemoglobin conditions. But problems or limitations with this method. Okay, is not every protein component like that gets separated. Let's imagine that the size is more or less same. You will not see the difference. They both overlap. They look at the same position. So not all hemoglobin variants can be separated, and also quantifying them. Normally they use softwares to quantify. It's like pixels. They normally quantify the pixels of this. They put a box around this and they quantify the pixels in this. So, but it is not accurate. It's rough approximate estimation they can do. So electrophoresis, although simple and quick method, is not very accurate method. Okay, that can be a limitation here that you can write. But otherwise, there are chromatography methods. One example that I would like to give you here. So because we talked about electrophoresis, one more method I'm going to tell you briefly related to hemoglobin, okay, or in general also. So several chromatography methods are used. Chromatography is a very, very common biochemistry tool. Chromatography method. So which is more sensitive than this type of electrophoresis method, can be used for detecting hemoglobin abnormalities in our body. Detecting hemoglobin abnormalities in our body. Excuse me.
So can you repeat the statement?
Yes. So chromatography, electrophoresis limitations we talked about, right? It is not so accurate. So chromatography methods are more sensitive. Chromatography methods are more sensitive, can be used as more accurate, tools for detecting abnormalities in hemoglobin. Abnormalities in hemoglobin. Because the hemoglobinopathies are the collection of diseases related to hemoglobin abnormalities, right? For studying them, electrophoresis is a simple tool, but not as accurate as the chromatography methods. Okay, right. So an example, like what is chromatography? I just mentioned to you like chromatography. What is chromatography? How does it work? What is the principle behind this method? So chroma means color. In chromatography, chroma means color and graphy means separation. So color separation, basically this technique means color separation. Separation of color, chromatography. So separation, what does it mean by color separation? Although originally this technique was used in separating colors, pigments. So you have plant pigments. So plants have different types of colors, right? You take the plant leaf, extract, and put it through this chromatograph. It's like a cylinder-like structure. There is some material into that. You put it through that. What happens? It would separate into different pigments. Pigments of different colors. So like that, traditionally, chromatography method has been used for separating colors. That is why it's called chromatography, the technique that is used to separate colors. But it can do much more now. Nowadays, I think you can separate the different types or different forms of chromatography methods. They work on different principles. But basically, the idea is they work like filters. They act like filters. Some of them are size-based filters. In electrophoresis also, we have seen size-based separation, right? So size separation happened, but here they are more fine filters than the electrophoresis. They can be more fine filters, and you can even change the size of the filters, size of the holes in the filters. So this is a method where more accurate separation can be done. As you see here, the size is blue colored ones you want to separate from the red ones. You see here, so blue ones come first depending on their size, and red ones comes next. So like that, size-based separation can be done for them. Okay, this is one method. This is called size exclusion chromatography or gel filtration chromatography we call. Gel filtration method or size exclusion method we call. There are many other types of chromatography methods. Some of them, what happens if you as soon as you add a compound to it, it will attach to the material in the container here, or we call it column. So like that, you can develop different methods, but in protein biochemistry, in the laboratories where they work on proteins, like hemoglobins, so these methods prove to be more accurate. Okay, and you can even separate them and study them further. Further studies can also be done on them. Okay. So based on this chromatography method, which is more sensitive and more accurate, you can even separate the product. Maybe you can separate phenylalanine from this. You can separate homogenesic acid, okay, and purify them and study them further. Their features, their abnormalities, their toxicities, those can also be studied using chromatography methods like this. So like that, hemoglobinopathies can be studied using electrophoresis or chromatography methods. So very common. Many laboratories even today use chromatography methods for studying various metabolites, including proteins, enzymes. And my field of research is also on this. I used to do a lot of these experiments to purify proteins, enzymes, and study their function, identify their function. But for our exam point of view and also for anthropology point of view, we don't need more technical details. We just need a concept. We just need an example of tool. Could you follow this? Could you follow electrophoresis and chromatography? I made a brief paragraphs of summary on this. Okay, if you want, I'll show you that. So this is what I'm going to share with you so that you can study. Okay. Just I'm pausing it for a minute. The handout that I'm going to send you will contain this information in it. Okay, students material. Yeah. So this is what I have made. I'm resuming this. So I made a summary, explained what is biochemistry, what is biochemical genetics. I briefly explained what are inborn errors of metabolism. alkaptonuria. And you don't need this one gene, one enzyme hypothesis. Just aware about this. I think I'll remove this part here. Just be familiar that initially in biochemistry, they talked about them. So this is not important for the exam point of view. Briefly, biochemical methods. I've explained whatever I discussed with you. Electrophoresis, I've explained it here. Electrophoresis method, I've explained it here. Okay, and also briefly mentioned about electrophoresis of proteins. I'm also going to remove this part. This is more specific, but I feel this is a bit complex for some of you. Instead of using this term, this is nothing but electrophoresis of proteins. I'll briefly leave out the component for knowing how electrophoresis is used for proteins. Just the way I explained to the size of hemoglobins, like that. Okay, then case study for biochemical methods in hemoglobinopathies. I've explained what are hemoglobinopathies, and also I gave some statistics that you may need here. So some hemoglobinopathies are more common in Southeast Asia and sub-Saharan Africa, given some factual data here. Okay, and then how hemoglobin variants like HbS, HbE are also a problem in India, and how electrophoresis method, hemoglobin electrophoresis method can be used in studying this and separating this. So separating them and quantifying them. Okay, and briefly I mentioned about chromatography method and the size-based as gel filtration method I just showed you, or I was talking to you. I've explained that, and how chromatography methods are very good tools for biochemistry studies. Okay, so right. So that should be sufficient for biochemistry part. There's so much that we can learn here, but I don't think they're necessary for the exam. That is why I've just minimized to this few specific things here. I wanted to show you this image as well. So using chromatography methods, there are specific methods as I was telling you, one something called HPLC method. You can separate even tiny changes, even if there are tiny differences between hemoglobins, you can separate them based on this chromatography methods. You see them here. So it's also like a filtration method. So what you notice here, HB1AC, HBE, HBF, HbA1, HbA1a. So many subtypes of hemoglobins can also be nicely separated with more accuracy. So like that, chromatography methods help us identifying these type of abnormal hemoglobins in our body, not only abnormal types of hemoglobins, abnormal quantities of hemoglobins in thalassemia can also be studied. Okay, right. So this is it. If you want, if you even feel this is more complex for you, explain the example of diabetes. Take the example of diabetes, how diabetes caused. So there you talk about blood glucose levels are increasing because of problems in insulin levels. Okay, like that explained. So it's also an example for biochemical problem. And how do you diagnose the problem? For diabetes, you diagnose the problem using simple biochemical tests like urine test or blood test can be used to specifically identify the location of glucose. But the techniques I have showed you here, bit more sophisticated techniques with a well-known example of hemoglobinopathies, which is also found in India. Right, this is a biochemistry story. Okay, biochemical genetic story. Next, I'll give you a brief overview, introduction to the immunological methods. So partly I'll do it today and partly we will do next class, in the next class, where immunological methods and recombinant DNA technology methods. It's the last part of this unit. We are going to deal with that. Okay, right. First, we follow the same approach that we took for the biochemistry part. Okay, so first of all, we try to define what is immunogenetics. This immunogenetics question had come in the exam before, not biochemical genetics. So far, they have not asked the question on biochemical genetics, but a question on immunogenetics have come. The question came like this in 2015. What do you understand by immunogenetics? What do you understand by immunogenetics? Explain it with suitable examples. So what is immunogenetics? Explain it with examples. So what is immunogenetics or what is immunology or immunogenetics? So you all know immune system, right? So our immune system is the system in our body which protects us from infections. Correct? Immune system is a component of our body which protects us from various infections. So like resistance that you have with respect to diseases, that is because of our immune system. Correct? So what do you learn? So what do you learn in immune system? How does immune system function? What are the various components of our immune system? How genes play an important role in our immune system function? These are the questions that you have to ask. What is immunity or what is immune system? What are the components of immune system? What is immune system? What are the components of immune system? Okay, what is the role of genetics in defining immune system functions? So here you are explaining what you understand about immunogenetics with examples. Again, immune system is also a huge field. So what do we learn in immune system function? You all know immune system, as I was telling you, provides resistance against various infections, diseases, even non-infectious diseases like cancers. We have resistance in our body with respect to it. But who runs them? Who are the integral components of, or what are the integral components of immune system? What does comes to your mind? What type of cells actually are the key players in immune system? Non-biology students, what are the key players? What are the key players in immune system function? Any idea? In general idea, what do you know about the cells that participate in immune systems?
WBC.
WBC, white blood cells. White blood cells are WBC. White blood cells are considered to be the policeman of our body. They always monitor our body for foreign particles. If they see foreign particles, they destroy them. And there are not one, there are multiple types of WBCs. Each of them detect our body. Each of them detect problems entering into the body and destroy them. So WBCs actually play the important role, most important role here. Okay, so study of WBCs, specific types of them, nature of them in providing immunity to us. Okay, and most importantly, some products of WBC. What are they? Antibodies. The most important aspect of the immunogenetics is learning antibody production. How do we get so many antibodies being made? So that is what I'm going to focus later. So mostly in the next class, I'm going to give you the scope here and a few terminology related to immunogenetics. Okay, so what are all important here? First, self and non-self recognition. I'm giving you a few concepts here about immune system function. So one is self and non-self recognition. So how do we know that some particles are foreign particles? It's like this. We grow dogs, right? We have dogs. How do dogs know that these people belong to home and these people are, um, like these people are actually new, that we have never seen them? There may be a threat from them. How do dogs know that? I'm not asking the mechanism behind it. Simple idea.
Well,
Let's say smell, let's say they see them, they recognize them, mostly smell that they use, right? But they're used to them, they're used to their smell or other components of them, but they're used to them. We see them. Similarly, in our body also, our immune system sees all our parts. There's some sort of screening, scanning happens when we are born. Okay, so anything that is new, that is not recognized by the immune system, is considered to be foreign. So like that, our immune system has ability to recognize self from non-self. So what belongs to our body, what belongs to, or what is not belong, what is new. So what is coming from outside the body. So our immune system has this great feature, is recognizing what is our parts, what are our proteins, our molecules, and what is new. So that is called self and non-self recognition. So this is to provide you the background related to immunity. In the exam, you don't discuss them. In the exam, you write something that we are going to build on top of this. Okay, immune system has self and non-self recognition. Did you understand that feature? It is being able to differentiate between what belongs to us and what is foreign. Is self and non-self. Second important component is antigen and antibodies. I'm sure you heard about antigens and antibodies. So what are antigens and what are antibodies? What are antigens? Which of them are foreign? Which of them belong to us out of antigens and antibodies? Can any one of you tell me what are antigens? We hear so much related to COVID-19 now. COVID antigens. So what are they?
Antigens are what the foreign body produces in our cells. And
Correct.
antibodies are what our body produces to counter them.
Correct. So antigens are foreign particles. Like bacteria enters into the body. Bacteria contains so many tiny particles, each of them can act like antigens. So bacteria has several proteins on their surface. They can act like antigens. Okay. So antigens are foreign particles. Example, bacteria, virus, so many. Even sometimes pollens, some people develop allergy, right? Because their immune system sees them as a foreign particle. Just because you actually inhaled some dust or some pollens, your body responds to it unnecessarily, causing allergy. So like that, even they can also be antigens. Like that, we have antigens being foreign particles, microorganisms, or maybe pollens, dust, hair of dogs, pets can be seen as antigens by our systems. And what are antibodies? When our body sees antigens, as you see in the image here, so these in the circular part is like an antigen or a microorganism on which you see orange parts here. So these are antigens. So when our body sees these type of antigens, our body makes these Y-shaped molecules called antibodies. We produce these Y-shaped molecules called antibodies. So antibodies are molecules produced by immune system. Write down. Antibodies are molecules produced by immune system against antigens. So antibodies are molecules produced by immune system against antigens. Okay, right. So study of antigens, study of antibodies are also part of immunogenetics. So we are going to focus on antibody production. This is, I think, the key theme in immunogenetics. How do we produce such a variety of antibodies against so many different types of pathogens? Understanding that diversity is a very, very important component of immunogenetics. Some students fail to understand it, they have difficulty in it, but I'll simplify this concept. I'll give you brief points, 4 or 5 points to understand immunogenetics. I also made a summary on this. I'm going to provide you this handout, so you'll be able to understand. So if question comes on this, you give scope of this and explain antibody diversity. What else we can have? So there are many genetic components. We're going to talk about several genes that are involved in antibody production. So genetic component of antibody production we're going to learn in the next class. Otherwise, we have several different types of WBCs. There's so many types of WBCs. I'm not sure if you heard about lymphocytes. So T cells, B cells, within them we have T helper cells, T cytotoxic cells. There's a lot of different types in WBCs themselves. The different types of WBCs. One such category is lymphocytes. Whenever we get infection, whenever we actually have an infection, doctors would ask for a CBP, complete blood picture. So in them you see lymphocytes, you see basophils, you see eosinophils. They're all different types of WBCs. And you always see elevation in their presence when we have infection because they are the immune system components. As soon as infection happens, they come into the picture to fight. That is why their level increases. Usually their number increases during infection. That is when doctors say, okay, you seem to have viral infection because your WBC count is high. Like that, they would normally utilize this as a immunobiochemical method here. By looking at the cell side, they can tell you whether you are going through an infection. Next concept is there are two different forms of immune system function. Okay, if you read this from class 12th NCRT, we have health and disease chapter in that. There they talk about how immune system functioning happens. There they discuss two aspects. So one, there are the two major types that we protect our body. Okay, two major methods in which we protect our body. So you all know antibodies. I just told you, you know antibodies. Antibodies are produced by one particular type of WBC called B cell. B cell is a type of WBC. B cell is a type of WBC which is responsible for making antibodies. Don't worry about this plasma memory cells. Just write down B cells and on one side show antigen. When B cell is exposed to an antigen, B cells produce antibodies. I just told you our immune system produces antigens against antibodies against antigens. Who produces them in the body? It is a particular type of WBC called B cells. So B cells are nothing but a type of WBC whose function is to make antibodies against antigens. But this is only one way we protect our immune system. Whenever we have bacterial infections, okay, or whenever we have something that is going in the blood, we make antibodies against them and destroy them, remove them from the body. In addition to this method, our body also has another type of lymphocytes or WBCs called T cells. T cells. T lymphocytes we call them, or T cells we call them. Our body produces also T cells. These T cells are responsible for another type of immune response. B cell is responsible for making antibodies. Antibodies are like bullets. You're shooting bullets at bacteria, you destroy them. You basically kill them. This is one method that our immune system uses. Second method is T cell based method. So in our immune system, we also have T cells, which are also other type of WBCs. What do the T cells do? T cells make some chemicals. They produce some chemicals. They don't produce antibodies, but instead they produce some chemicals. And what do these chemicals do? In this analogy, I told you, antibodies are like bullets. Chemicals are like explosives, are like bombs. So they produce lot of chemicals. Lot of chemicals are made. These chemicals destroy cells which carry the pathogens, because viruses like HIV always hidden inside the cell. You would not see it in the blood, because if it is seen in the blood, we make antibodies using B cells. But some of these viruses they hide inside the cells. What do? The cells actually give you an indication. Cells say, oh, I'm in danger. My body is affected. Cell will give that signal to the immune system. The immune system recognizes them and produces chemicals. What do these chemicals do? These chemicals blast those cells. Okay, so they defend against infected cells, or cells where pathogens are hiding inside them, which is a common theme with viruses. Some viruses, they hide inside the cells. And also these T cells are responsible for detecting cancers and preventing cancers from happening. In our body, we have a mechanism, we have ability to prevent cancers, but it doesn't always work, but we can protect ourselves from cancers. We have inbuilt ability for that. That is also done by T cells. So like that, we have two types of immune system responses. One is mediated by B cells. So you can call it antibody mediated immune response. Another one is led by type of WBC called T cells. Don't worry about types of T cells. If you read it somewhere, the two types of them, helper cell we call, is what HIV targets. HIV is such a troublesome infection because HIV targets a key component of immune system. It attacks T helper cell. So if it attacks T helper cell, what happens? T helper cells collapse. Our immune system collapses. And that is why in HIV, when it becomes AIDS, we are prone to more and more infections and more problems, including cancer, because the activity and function of T helper cell is gone. But otherwise, how do T cells act? T cells act, especially this T cell is the active T cell. This T cell acts by producing some chemicals. Nowadays, you also hear about this in COVID-19. COVID-19, they talk about not cytokinases. What is that? Cytokine storm. You heard cytokine storm that comes in the newspapers related to COVID-19. So there, cytokines means chemicals. I'm telling you the chemicals released by T cells. Such several chemicals are released in COVID-19 infection. That is why people are prone to more and more issues, not just because COVID-19 virus got into our body, because our immune system responded too much to it. So several people, especially those in the old age, seem to be dying from COVID-19 infection more than the other people because of over signaling of immune system. Okay, or overproduction of those chemicals I was mentioning to you. These chemicals are called cytokines. Cyto, C-Y-T-O, cytokines, K-I-N-E-S, cytokines, C-Y-T-O-K-I-N-E-S. Cytokines are chemicals that are released by them. Okay, right. So this is a brief idea about immune system function, immune system component. This is a very basic level immunity component, which is also important for your prelims part. of the exam, because some basic questions can be focused on something like this. But for us, I wanted to give you a brief idea on immunogenetics and what is immune system, and overview about immune system. So far I told you this, that immune system is the protection mechanism of our body, and it can recognize self and differentiate it from the non-self or foreign particles. And we have antigens or antibodies. Antigens are the foreign particles that enter into the body. Antibodies are the molecules or the products of immune system made against the antigens. And genetic components for detecting antibodies, for producing antibodies, we have some genes that are responsible. There is a very complex game here that we are going to discuss in the next class. And we have different types of WBCs. We talked about B cells, T cells, and how B cells make antibodies, and how T cells are responsible for cell-mediated cytokine-based immunity. Okay, right. So also other examples that you may need to use for this immunogenetics is blood groups. Even detecting blood groups, presence of some proteins on the blood groups can be discussed under immunogenetics, which we already talked about, because blood groups contain antigens on them. Correct? We talked about A blood group, A antigen, B blood group, B antigen, AB blood groups, AB antigen. So antigens are part of immunogenetics. So blood group studies can also be discussed as part of immunogenetics. And then the most importantly, HLA. These two examples we are going to cover. One is antibody diversity, another one is MHC or HLA. This is also part of markers, another unit, 9.5, I think 9.5 or 9.4. So markers, unit on the markers. So 9.6 indeed. So there we are going to learn about HLA. This HLA is also very, very important component of immunogenetics. So tomorrow I'm going to cover these two things. One is antibody diversity and HLA. Like today, I'm also going to give you an example or two of methods. But you can also take the overlap. In biochemistry, we talked about electrophoresis. That can also be used as a method in immunogenetics. So if you want to reduce your burden in studying that, you can go with electrophoresis, but take example of immunogenetics for using electrophoresis. And for biochemistry, just talk about hemoglobinopathies. Okay, so tomorrow we'll cover those two aspects of genetics under immunogenetics and diversity of diversity and HLA and a method or two. Right? Okay, so tomorrow we'll be able to finish this and move on to new DNA technology. I don't know whether we'll be able to finish recombinant DNA technology, molecular anthropology. Okay, so that is something that is routinely coming in the exams these days in the last few years. Right. Okay. Any questions, guys? Any questions on this before we are done with today's session? Are you able to follow this, or was it looking too complex for you? Is this digestible? Or what we talked about today is simple enough for you to follow? Unsure? Shweta, Sunil?
It is okay.
It is. Okay, Shweta, what do you say?
It was okay, but need to study the material.
Correct. Yes. So if you want to read more on this, biochemistry, I don't think there will be any specific paper article talks about. Biochemistry is a huge field. What I've gave you is a small glimpse of it, summary of it. So read the handout I've given to you. Focus on that. Okay. Otherwise, there's nothing specific for that. I think Britannia Encyclopedia has some information which I used as a reference for this, but otherwise not much resources on that. Because it's a huge field. You will find so much, but comprehending it and summarizing it is challenge. I don't know if IGNO material has anything on biochemical genetics. I have to see that. Okay, but otherwise read my handout that I'm going to post you right after the class now. Read that handout and see if you have any problems there. Okay, short note. If you get immunogenetics, biochemical genetics as short notes, what do you write? Examples, brief summary. Only prepare so much. That should be enough. Okay, right. Okay, if no further questions, yes, I'm going to stop the session here. So is 4 o'clock all right with you then? Is the 4 o'clock time is all right? Okay, right. So we'll continue at 4 o'clock. We'll take the class at 4 o'clock tomorrow. Bye, guys.